Good morning, everyone. Welcome to CCNEP. Uh, so, those who are first time here, this is our weekly kind of meeting organized by the organizers. This session, we are very good and lucky to have Raisha Aidin, our new introduction afterwards. Some housekeeping things as usual. In this new normal, please mute all your mics and if you have any questions, please put it on the chat box. Place questions which answer later at the end of the talk. So a little bit about Dr. Noraisha Maidin is our speaker today. Uh, Dr. Noraisha comes from a very good department or unit, longest name you can ever hear, invertebrate and vertebrate neurobiology laboratory. If you have not heard of that, that's wrong. Uh, so she's a very remarkable person whom I have the privilege to know. And I think that maybe just 5 years or 10 years. Actually, it goes back 30 years. But I want to say a little bit after that, because as a patient, uh, I'm proud of her that she team layer. So she graduated uh, PhD. In fact, her degrees are remarkable because it ranges from basic science to anthropology. And
Prof,
the
I
latest one
think
was
you need
a
to ask
PhD,
your, sorry,
on
Prof,
developmental neurobiology.
sorry,
Sorry?
I think you need to ask your fellow, attendees to switch off to mute their microphone because you're not coming out clearly. Yeah, can't really hear you clearly.
Okay. So I mute everyone now, yeah? Can you hear me now, Aisha? You
Yes,
can,
yes,
huh?
I can hear you. I can
Okay.
hear you. You're brilliant. Yeah, great.
Okay, good. So, back to where I was. So, Dr. Raisha has many, many qualifications, including on entomology, as well as developmental neurobiology, which she got a PhD from the University College of London in 2007. She joined University Malaya in 2008, I believe. And I remember the day that she dropped by the PITS department way back in 2008 or 2009. And she has been going up and up ever since. But on a personal note, I know that she has been very active in research. She had many, many grants, many publications. But more than that, she is a strong-willed person because she has spinal bifida herself. All right, and for many reasons, she has been a champion, a mentor for many people, and also activist, if you like the word. She's also the secretary to the Persatuan Academic, yeah, Kaki Tangan Academic University Malaya, and she has been very vocal in many instances. And she has been on many talk shows, and she has her own website and so on. So, but more than ever, I think she's very passionate about the topic on neural tube defects, and that's why I have her here today to educate all of us on this complex topic. Many clinicians think that NTDs are old story from the past. It's not. It's a big problem. I think we need to really refresh our knowledge on this issue. And there's no other person that I know of who can talk more about this the whole day than Dr. Noraisha Maidin. So please welcome this great lady who will talk to us about neurotube defects, animal models, and recent advances. So, Aisha, your turn.
Yeah. Okay. Thank you so much, Prof, for the very, very kind introduction. Hi, everyone. I'm Aisha. I've been in UM for some time now. I am a parasitologist, parasitizing the parasitology department. I have three labs there, and collectively they are called the invertebrate and vertebrate neurobiology lab. Yeah. So everyone is welcome to come at any time. I'm happy to have you, and you are able to WhatsApp me if you want to set up an appointment, if you want to have a look at the lab and the equipments. I'm very, very happy and open to collaboration. All right. So coming back to why we are here this morning, yeah. Um, Neural tube defects, as Prof. Tong said just now, it's not an old issue. It is actually a major issue in Malaysia. But not much is talked about it because there aren't many research groups working on neural tube defects in our country, right? So oops, my slides not working. All right, okay. So now, this is a heat map, yeah, that I want to share with you to show the distribution of neural tube defects globally. All right. So we have stressed that neural tube defects is not an old issue. It is not something which has been sorted out because we found folic acid, yay, we have to do the job and that's it. It's not. I will illustrate why, and I hope in the next 30 minutes or so, you will have a better understanding, and you're more than welcome to interject at any point if there are questions, or keep it to the end. Yeah. Okay. So in terms of distribution, there's only two papers that's been published on Malaysia, and you have uh, the first one was published in 2013. By Prof. Tong and team. The second one was, together with my team, Prof. Tong as well. Um, so the first paper that was published in Tropical Pediatrics, if I'm not mistaken, shows that the prevalence of neural tube defects based on the birth defects registry in Malaysia is 0.42 in 1,000 live births. All right. So this is the number, and this number is actually low in comparison to the global occurrence, and the accepted global occurrence have always been 1 in 1,000 live births. But now, interestingly so, even with the advent of folic acid, you find that most review papers now quote the numbers globally at 1 to 10 in every 1,000 births. All right. So one of the things which I have done is to rifle through the data in Pusat Perubatan Universiti Malaya, for example. Yeah. So we had published that in 2017. And that was actually brought upon largely because I also run a family support group for spina bifida, individuals with their families. And I often get calls, right? So looking at just the data itself, we find that there is 2 to 9 for every 1,000 cases reported. All right. And this was looking at 13 years worth of data. We have recently gotten human ethics approved at three other hospitals, UKM, HSA, as well as HKL. And we hope that, with the four hospitals together with we will be able to come up with a more up-to-date, information on what is actually happening among patients with neural tube defects, specifically with spina bifida, as my team rifles through every single patient record that comes under the ICD-10 classification of spina bifida. All right. So we can talk more about that later, but what is important right now is to look plainly and bluntly at the Malaysian scenario. All right. Okay. So we know that it is, the most common central nervous system malformation in humans. Second leading cause of birth defects next to heart defects. Second leading cause of disability next to cerebral palsy. If you look at the health economics papers, obviously the paper that has been quoted the most is the one from the United States, and that one showed a very Ying et al., if I'm not mistaken, showed a very comprehensive breakdown of expenditure for myelomeningocele for just 20 years of life, and that was, it came up to 1.4 million. Uh, that is, super high, and that was published, I think, in 2011. That was 9 years ago. So we do not have in Malaysia, we do not have a health economics paper out yet in regards to neural tube defects. We don't know how much it costs, but what I do have a rough estimate, which Prof. Dharmendra had kindly allowed me, to calculate with him, is that looking at the numbers of NTDs in Malaysia, putting it in at a very conservative number. If we were to say, 1 in 1,000, there's 1 in 1,000 in Malaysia, and if you say half the numbers of that, have spina bifida, the other half have anencephaly, just a rough estimate. And even if you put half of those numbers, unaccounted for, so that would be over, I think, 7,000 people with just spina bifida. And if you were to take it that all these 7,000 were spina bifida aperta, and they were closed at the first day of life, um, and the numbers that we have already spent, the very minimal number is already at 63 million, to close the bags, to close the open neural tube, open spinal cord of the patient born with spina bifida aperta. Okay. Furthermore, Malaysia is not participatory to any folic acid fortification program as of yet, and we may not, practice in utero surgery for many reasons. There's only 5 centers globally that do it, 4 in the United States, 1 in Europe, and now more centers are opening up in the United Kingdom, where they are not doing, the open surgery. They are doing it laparoscopically. So this is a new technology pioneered by University College London. We're still waiting for more papers to come out from the, in utero rescue laparoscopically. But there are dangers, as we know, in regards to in utero open surgery. Yeah. Okay. So we also want to know, why is it, my group, that is, we are looking towards publishing a review paper at what is happening on the ground. And I think this is extremely important, because we need to know, how many babies out there in Malaysia, whether they are getting the kind of care, that any child with spina bifida deserves. I mean, in utero surgery, for example, has been around since 1995. I myself was born in 1975. So we need to do a study of, what is happening in our country. Because as I mentioned just now, the family group that I'm running, and because I'm active on social media, I get calls from all around Malaysia, whether or not it's from Sabah, Sarawak, Kelantan, Terengganu, Johor. People are calling me on a weekly basis, because they want information, they need information, they want to know who the clinicians and the surgeons who can look after their children. And of course, as these children grow up, like I have at least two spina bifidas in their 60s in my group, and they're doing well health wise. The lifespan of people with spina bifida are increasing in Malaysia, and people have questions. I have many young individuals who are at the cusp of, the job market, and they come to me again, questions in regards to, life and maintenance, and how I get about, for example. All right. So now I'm going to go into the science, as to how it is, that I right how it is that I got into the science, or the biology of NTD. Okay. So I applied, actually. I applied to Professor Andy Cobb and Nicholas Green's lab, way back, in the early, I think 2000. I think I left for England December 2002, if I'm not mistaken. Yeah. And that was when I was first exposed to vertebrate models of neural tube defects. So if you're looking at the neural tube, if you're looking at the fish or the frog or the chick or the mouse, these are all perfectly good models to study developmental biology, which can be used to study the neural tube. But the argument that is put across by Professor Andy Cobb is that the reason why we use the mouse is that um, if you were to section the mouse embryo, let me show you what a mouse embryo at the beginning of neurulation looks like. All right. So I hope you can see my arrow here. Yeah. So this shows the developing brain. All right. This shows the soma is these blocks. The blocks are the presumptive um, muscle as well as the presumptive vertebrae. This is what it will develop into, and this is the open spinal neural tube, or what we call the open posterior neural pore. Okay, so this is actually the point where we're really interested in. So in the mouse, rate of development for closure of the neural tube takes 3 days, day 8, day 9, and day 10. So this is an early stage day 8, which is akin to the human development of the neural tube between day 16 to day 32. You find that the two sites meet up together in a 6 to 7 somite embryo, and then the closure will proceed rostrally in order to get a complete closure of the brain, and it will also proceed caudally for the entire spinal cord, presumptive spinal cord to close as well. All right. So these electron microscopy images, these three actually, these are all my images. I love EM because it really gives you a clear look at what's happening. So when I questioned my supervisors in England, why is it that we study the mouse? Yeah, uh, and it was very interesting. I love it when professors show me data, like data that they have obtained with their own hands. And Andy showed me a series of pictures of a human neural tube undergoing neurulation, undergoing the process of neural tube closure, and he showed that every step of the way was similar to that of mouse development. Okay, what that means is that schematically, this is how it looks like. The picture in A shows the orange bit is what you call the surface ectoderm or the non-neural ectoderm, and this it undergoes this process called convergent extension. So the two sides basically come closer to each other, and it also elongates anterior posteriorly to form the tube. And then you have this structure called the median hinge point right in the middle here, and the notochord is down here. It will elevate and then it will bend. Okay, it will bend until it comes to opposing ends, and these opposing ends will culminate in what is known as adhesion and fusion. And that was what I was realistically looking at during my PhD. What are the requirements in order for the two sides to meet together? So Andy, my very, very interesting and well published PhD supervisor, Andy broke down development in such a way that he looked. I mean, no one else before him actually looked in the mouse neural tube. What are the requirements for convergent extension to happen? What happens when the convergent extension fails? And Andy, went further by also looking at mouse models which, if they fail at convergent extension, what would the phenotype be like? And then followed on by failure of elevation. What happens when there's a failure of elevation? Andy was also of the opinion that these dorsolateral hinge points, this is what he calls them, were a unique event. And but again, the thing that I love most about professors, especially when I was in UCL, is that they're always open to scientific debates, and I argue that this is not a gene driven event. Convergent extension, you do have genes expressed here, elevation, also you have specific genes expressed that are not expressed during convergent extension. And dorsolateral hinge points, however, does not have a specific gene expressed here and here, for example. So these are the things that makes breaking down every developmental time point in a model, in a vertebrate model, crucial for the understanding of what is happening in the human. All right. So it's very, very robust data, and I have the highest respect that a human, Andy managed to get a human fetus undergoing neurulation, and he showed, look, Aisha, look, it is there, convergent extension does happen, elevation does happen, dorsolateral hinge points does happen, and the adhesion and fusion also does happen. So it's very, very evidence based and evidence driven. So when I came back to Malaysia, I thought, oh my God, like developmental biologists publishes so much so fast. How on earth can I possibly compete? And so I decided to look at something that, say, the rest of the world were looking at, and having spina bifida myself, obviously, I have high respect for the clinicians. I have a high respect for the surgeons, the very, very talented surgeon who have kept me alive and well and functioning over the years. And so I wanted to know what are the things that affects them, what bothers them. And of course, not many embryologists out there are actually looking at the difference between the spina bifida aperta model, which is the open version, as opposed to the spina bifida occulta version, which is the closed version. So I've had numerous, debates and arguments with both Andy and Nick in regards to the etiology of the occulta type spina bifida, and a lot of which that was mentioned, over the years is that if you were to compare it to the mouse, spina bifida occulta was likened to the development of the mouse's, sorry, I don't know whether that's correct English, the tail, the formation of the tail. So it was, in a way, on an embryological viewpoint, occulta was rather poo-pooed, or not looked at very, very seriously. Because the idea was that it arises from failure of secondary neurulation. So I then pointed out that in the clinician's viewpoint and in the surgeon's viewpoint, irrespective of whether or not the underlying neural tube is open or open or not, obviously an embryologist like Andy would call open neural tube as aperta. And then I counter argued and I said, no, well, clinicians and surgeons will say that if you have a skin covering it, they call that occulta, they call that the closed version. Even though underlying the closed skin is an open neural tube which has developed into neurologically impaired spinal cord. So that was my argument, and the other argument was that I argued that it doesn't matter how the level of the lesion as well. The level of the lesion can be very high in a spina bifida occulta child. So I have seen progression of review papers that have softened over the years, and I think it is very crucial that the embryologist works together, the scientist works together with clinicians and with surgeons in order to find the common ground in figuring out, the devastation that we know, which is neural tube defects. I mean, if anyone has managed a child with spina bifida, have seen the look of panic on a parent's face when they first see their child's back, their child's, exposed spinal cord, will tell you that it is not an easy condition. This is not an easy condition that we're dealing with, and we need all the arsenal that we can get, including especially by using animal models. Okay, so I'm just gonna dive straight into it. Why are we using these mouse models, right? Because experimental studies obviously cannot be performed on human fetuses unless you're planning to go to prison. Yeah, so uh, we know that has happened. Scientists who are now looking into CRISPR-Cas9 technology for and using it on human. Okay, sorry. Hi. Okay. Okay. So now, experimental studies is one thing that you can do on the mouse model. Uh, Understanding developmental mechanisms, looking at the cell biology, uh, you know, these fancy experiments in introducing whether you're looking at inhibitors or whether you're trying to rescue the phenotype. These can only be done in animal models. But the best thing, I think, in my opinion, is that you want to find a way in order to prevent this condition. And there is no better way than that of looking at dietary rescue. All right. And this is further compounded by the fact that we still do not have a neural tube defect gene which can be used to screen the global population. It's ongoing in different cohorts of the world, but it is not happening in a sometimes I wish Barack Obama is still the president of the United States, but maybe AOC will become, and that will then drive towards, a bigger, a global initiative to find the human NTD gene. All right. Okay. So another reason why mouse models are very, very crucial and very, very good to be used in understanding the formation of the neural tube is that when you harvest the embryos during development, yeah, and we do that regularly in my lab, you can see failure of convergent extension leads to craniorachis. This is a mouse embryo. You can see the exposed brain. You can see the open spinal cord, and this is very easy to see when you are dissecting. Even though the embryos are super tiny, mouse embryos are anything between half a centimeter long, and they're super tiny, but you can observe the phenotype very clearly, of the open brain and the open spinal cord, and this is commensurate, or this is comparable to what we see in the humans. Okay, so failure of site 1 leads to craniorachis. Failure of sites 2 and sites 3, the midbrain here, the developing midbrain, and the developing forebrain, will lead to the condition that we all know is called anencephaly in humans, but we call it another name in mouse development. We call it exencephaly. Again, very obvious and very easy to see when you're dissecting and you're harvesting the embryos. All right, and so if closure caudally to the open spinal neural tube does not complete, you will get the condition known as spina bifida. And if you see an embryo past day 10, if you still have an open posterior neural pore, you know then that the neural tube will not close, irrespective. Okay, so I mentioned just now that the one thing that you can control in order to prevent neural tube defects from happening is the diet. All right. So when we're talking about the diet, what kind of diet is it that helps for the neural tube to close successfully? All right. So that is what we call the perinatal period, okay, which is the beginning of pregnancy, and well, it's actually during the pregnancy. So this was a slide that was drawn by one of my students, and we are very, very interested in looking at perinatal diet, um, because you can score the mouse embryo phenotypically, and also because we really need a public health policy to understand environmental factors which triggers neural tube defects. And diet is a good option because we can control it or attempt to at the very least. Okay, so my lab focuses on this relationship between mouse neural tube development and their relationship with the perinatal diet. And we're going to show you. I'm going to show you some very very interesting unpublished data. But before that, I think it's important to mention here that um, that there's many reasons why we look at diet. Folic acid being the most obvious one. All right. I will also tell you a little bit of history in regards to this. Um, but mostly, maternal nutrition. It plays a major role because it is an important determinant of embryonic and fetal growth at the time of conception and the first 2 weeks of pregnancy, which includes the pre-implantation period and the rapid placental development, which is highly dependent on maternal nutrition. And I'm going to show you how we do that, how we study that in the human. All right. But before that, I think everyone I think should appreciate what actually happens, the history of you know well, we'll talk about folic acid because it's the most famous dietary vitamin available in order for the prevention of this condition. All right. So I need to introduce you to these three gentlemen. The one on the leftmost is Nicholas Wald. All right. So Nicholas Wald is the clinician and scientist who came up with the MRC, the 1991 MRC vitamin trial. And he was the one who said, like, if you were to take folic acid, you can prevent 70% reoccurrence of neural tube defects. One of the first things that I came across when I first went to England was that I found out that my supervisor, Andy Cobb, does not get along with Nicholas Wald. All right. So it's very interesting that all these very, very hardworking people who are championing their science, and they do it ever so passionately, and they argue, and I think it's very, very important that science is balanced by both sides of the argument, right? So Nicholas Wald obviously said, okay, I give this amount of folic acid, 0.4 milligrams to the mothers, and 70% of them, if they've already had an NTD baby, their babies will be rescued, right? So it takes someone who is as brave and as hardworking and a little bit out there, for Andy to argue, well, what happens to the other 30%, right? What do we know happens if we were to take, folic acid continuously for many, many generations? Those answers are only now slowly coming out. There are more and more publications. Yes, folic acid does protect, but at the same time, there are fears whether or not folic acid taken over the generation can promote cancer in the public, whether or not it can even cause autism, for example, because elevated blood serum of folate has been shown to have a higher incidence of autism in society. And so this is where public health policy cannot be a debate which is being done only by a select few. It must continuously be argued and be debated by everyone, and especially the stakeholders. And one of the things which I said when I went for my first Neural Tube Defects International meeting way back in 2005, I said, shame on all of you, shame on all the scientists, I said, because you don't have a single stakeholder group, you don't have a single family group or a neural tube or a spina bifida group represented at these meetings. And this is where, the gentleman on the rightmost, Godfrey Oakley, even though he is not well liked, I think, in my opinion, by the scientists at the NTD meetings, but his role is crucial because Godfrey Oakley will give his talk every 2 years at the NTD International, and at the end of it will flash a photograph at a wedding, and his gift to the bride and the bridegroom would be a bottle of folic acid. You know, such was the determination of Godfrey Oakley, who works with CDC Atlanta, Georgia, in order to make, you know, the whole country protected, to prevent children having to sit on wheelchairs. And he has even cornered me and asked me, why is it that Malaysia is not 45? So yes, I am honored to be in the presence of very, very passionate people in science, and I see the importance of understanding everyone's viewpoint. And I think this is something that Malaysians, irrespective of whether we are, you know, doctors, scientists, nurses, stakeholders, we need to have more, face-to-face discussion with each other, and try to understand where everyone is coming from, in order to help Malaysians. We really need that, yeah? Okay. Uh, Even then, okay, I thought this slide is very important because I wanted to show you the picture of Dick Smithers. Richard Smithers really is the gentleman who um, did a study on multivitamins, and he showed that, you know, without multivitamins, a person's pregnancy is far more a danger than with multivitamins. And this was followed, this idea was followed through by Nicholas Wall, who then targeted the folic acid. All right. Now, okay, this is another bit of highly interesting information. All right. There are hundreds of mouse models out there, all right, and all of which you can read. I've given my slides to Prof Tong. If you were to click on this, you will find over 250 mouse models with neural tube defects, and you will find that not all of these mouse models respond to folic acid, which suggests, you know, that it is similar to what is happening to the humans. That, you know, what happens to the other 30%, and I will talk about that. But before that, just looking at, you know, the various mouse models, I'm just giving you three different examples here: the LERP2 mouse, the splotch mouse, the cited2 mouse. They all respond to folic acid, meaning that given folic acid, there will be a reduction in the occurrence of the neural tube defects. All right. Okay. So you might also ask me, why is it that this mouse models only number 250 as of 2010? Why 2010? It is because the two most hardworking women in the field of neural tube defects, Muriel Harris and Diana Jurilov, and they would painstakingly, at every single international neural tube defects meeting, put up a mahjong paper, and get people to write, or to note or to state any mouse models which they have missed out, because they came out with review papers every few years. And it was just that they have both now retired, and no one is now, you know, holding on, the baton. It's a very heavy baton. So an NTD paper citing, all the mouse models post 2010 is a highly sought after publication. And I wanted to talk to you a little bit about Irene Zon, because Irene is among the younger generation of scientists, and she published this with the LERP2 mouse, and she showed that, you know, with the one carbon cycle, the LERP2 mouse, for example, can only be rescued. The exencephaly in the LERP2 mouse can only be reduced if the folic acid is given intraperitoneally. So this, you know, ties in together with the understanding that the folic acid pathway, or the what you call the one carbon cycle, is complex. There are different ways in order for the folic acid to be taken in and utilized, and this is something that we don't understand in the humans. And you can't really have a proper targeted approach for folic acid unless you do all this very minutiae of experiments to find out what really works and what pathways work, so that we can also have better public health policies to protect others, because we know that too much folic acid in the diet is going to be bad for the aged, because it masks pernicious anemia. And for many years, England did not fortify. It's only recently been fortified because of the lobby group by Nicholas Wald and son, George Wald, who's a cardiologist. So you have all these different groups doing different things, but it does not. But it even highlights how important it is to give funding to the scientists to figure out all these pathways. And just so that you know, there is data on Malaysia. This is a paper that was published in my group, which clearly shows, how we have used a drug that was given to a patient born in PPUM 7 years ago, and this child was born with a huge cyst, which was diagnosed as cranial meningocele by Prof. Dharmendra, specifically parieto-occipital cranial meningocele. And I didn't use any of the human data. We just focused on what was happening in the mouse, but we used the exact same tablets, which we had tested in 2 different places, at the National Pharmaceutical Bureau as well as the Pharmacology Department of UM. And we got this published in 2017, and we showed that in the mouse, it was crazy. I was opening up all these, during dissection, and for every litter, and the litters were quite large. There were like 11 embryos, and 4 out of 11, each of these litters had massive exencephaly. So it was a clear cut, evidence that mouse experimentation is critical in the understanding of central nervous system malformations, like neural tube defects. This family was able to get some kind of social justice, because they brought that private clinic to court, and they settled out of court. And I'm happy that I am helping to contribute to Malaysian data, so that we can have better care for our population. We need to do this work because no one else is going to do it for us. We need to understand what is happening in our population, and so this is a very it's a very clear clear evidence to show that the use of folate is needed in the prevention of neural tube defects. All right. The other thing that I also thought was highly interesting, and that I wanted to talk a little bit about, is because the mother, in this case, is an educated woman who held a very good job, and she was taking her folic acid. But it was just that, she was also taking an antibiotic, which is a folate antagonist at the same time, right? So that is an indication that some kind of rescue had also happened, because the child was not open with an open cranial neural tube. And all the mouse data that we did with the same drug showed that the mouse had an open neural tube, and when we tried to rescue with folic acid, there was just a lot of resorption, which shows that in the animal kingdom, if you can't survive, then resorption will occur, and so the embryo, the fetus, does not come to term. The other thing that I wanted to show you here is a mouse model that was created in my lab, also with a closed cranial neural tube. Uh, you know, we can do. I want to demonstrate here that we can do high quality science in Malaysian labs. Provided that the passion is there and that the funding is there, and this is possible. Yeah. Okay. So apart from folic acid, there is also a second type of supplementation which is being studied a lot, which is called inositol. All right. And how did the inositol study came about? This was actually a PhD project done by Nick Green. Nick Green and Andy Cobb were both my PhD supervisors when I was in London, and for the longest of time, the curly tail mouse was thought to be the human representation the mouse representation was thought to be the mouse representation of human spina bifida, because it does not respond to folic acid. I mentioned just now the models that respond. This is one which did not respond. No matter how much folic acid you gave it, whether you give it by diet, by intraperitoneal injection, etc., you're not going to rescue curly tail. You will have the defect in the curly tail mouse. So obviously, the hunt for the gene, for the curly tail gene came about, and this is where it gets really interesting with neural tube defects, because we know neural tube defects is a multifactorial condition. You can't just look at the genetic factors without looking at the environmental factor. And this is where it was really, really cool, because Nick published in 1997 in Nature Medicine, a huge achievement when you're doing a PhD and you publish in Nature. And he showed that inositol, specifically myoinositol, given to the curly tail mouse, will help with closure of the neural tube. And they did rescue the spina bifida in the curly tail mouse. But at that time, they did not know what gene causes the curly tail phenotype. So it's very interesting that years later, while I myself was doing my PhD in Nick and Andy's lab, that there was an Australian group working on transcription factors who just happened to knock out a gene called the grainy head like 3 gene, and they found that this gene was the curly tail gene, and this was the gene, the grainy head like 3 gene was the gene that causes spina bifida. And I want to interject here because I think the human factor is very, very interesting. The scientist that was working on the curly tail mouse for many decades, looking for the gene. Yes, he was mapping the gene. This Dutch guy, Henny van Straten. He retired. He retired as soon as this Nature paper came out by the Australian group. It is. Scientific work requires so much passion, and you really need to just enjoy the rigors of the science, irrespective of what you publish. I think in order to do meaningful work, okay, and I want to show the meaningful work that's being done by my team. I'm very proud of my team. I'm very proud that I have Prof. Tong in my group and many others, many talented scientists as well as clinicians and surgeons. Okay, I'm just going to go very quickly because I think I've talked for quite a long time. These are all statistical data to show that the black seed oil had a very positive outcome. It was a very specific type of black seed oil. Not all the black seed oils which we tested gave this result. And it was a very specific breakdown of essential fatty acids, right? Again, I still cannot understand why funding is so difficult to get from the Ministry of Higher Education. But yes, that is what is happening. We have published this bit of data, which shows coconut oil really, taken throughout pregnancy, during pregnancy, and after pregnancy causes low birth weight. And this was actually published in BMC Alternative and Complementary Medicine not too long ago by my team, which clearly shows that coconut oil may be good for the adult if you want to become slim and you want to diet and you want to look nice and pretty, but not when you are planning for a pregnancy. And this kind of education is not happening in Malaysia, and that is really, really worrisome for me. Okay. So I mentioned just now the two ladies, Muriel Harris and Diana Jurilov. Please do have a look at their publication if you want to. Their review papers are fantastic. They will list every single mouse model there is, which one responds to folic acid, which ones do not. And I'm sure once Nick and Andy, have more data globally on inositol, that paper will come out, and we'll also have show mouse models which respond, a list of them to inositol. And also another candidate that's coming now is the formate, and all of this work within the one carbon cycle quite differently, and it's all highly interesting. In fact, with inositol, for example, um, we it is good to be taken by diabetic mothers because it just makes the mother more sensitive to insulin in the blood. Yeah. Okay. There's just a few more minutes left. I am going to end my talk with a mouse model, that was created in my lab. And the mouse model that was created in my lab is a mouse model with spina bifida occulta. The picture on the rightmost here is a pup which came from my lab, produced in my lab. And you can compare that with the Aperta model with the open, exposed spinal neural tube. All right. And how did I, come up with this idea was because I studied the Fs and ephrins during my PhD. Again, we always look at nature papers. This was published in the year 2000, and it showed how, two very specific genes in the huge F and ephrin family causes exencephaly. I looked at the candidates in the spinal neural tube. And so, interestingly, I was trying to knock out the ligand when I was in England. I had to come back because I didn't have time, but maybe that was a blessing in disguise. Because when I came back to Malaysia, I bought the two receptors, the two knockouts, and I crossed them together. And lo and behold, we published this not too long ago, 2017, if I'm not mistaken. And we showed that it has not only a closed exencephaly, a closed cranial neural tube defect, but it also has a closed spinal neural tube defect. All right. And we hope, that we are able to continue procure funding in order to try to rescue. Because if we don't do the experiments, we won't know whether our FA2, FA4 mouse model responds to folic acid or inositol or formate or black seed oil. We won't know. Without funding, we cannot do these kind of important scientific work for the benefit of our population. All right. Mouse models, again, can be used to now rescue stem cells. This is a study that was published just this year. It just came out. By using stem cells, specifically bone marrow derived stem cells, you can rescue the rat neural tube defect. This is a rat. I am telling you, it can be done in the mouse. I have done it in the mouse. This is how it looks like in the mouse. You can inject substance in the mouse, even though it's a far smaller embryo. In my lab, when feeding experiments, we give the feeding in a humane manner. We do not force feed. We're just holding the mouse, and the mouse itself is sucking onto, in this case, retinoic acid. Retinoic acid is a chemical model which we have developed in the lab, and it does cause spina bifida. When we do data in the lab, tiny embryos, when we harvest the embryos, we measure the cranial neural tube, we measure the posterior neuropore, which is the spinal neural tube, we measure the number of somites, we also measure the crowd rub length. So all these data Raw data is extremely important in order to elucidate and to compare with the controls what is happening in the spinal neural tube. All these pictures were taken from my lab. We have the equipment. There was multiple HIRs and research funding that I have killed myself over the years, but it's well worth it. It's well worth it because, in my opinion, I want to understand what's happening in the human population in Malaysia. It is also important, I think, and I use this picture to educate my students, is that with mouse studies, you must also understand that there's a difference in strain. Certain strains tend to resorb. So you see this darkened bit, embryos which did not develop. So you will have a lesser number in the mutants than you do in an animal model which is not genetically or chemically perturbed. This is an ongoing paper with Prof. Tong investigating Sex63, and the candidate gene was based on exome data. This is a representation of the gene and the protein that was taken from the exome data in a Malaysian population. So this is a Malaysian paper, and then we try to translate that by looking into what happens in the mouse. So I'm ending today exactly at 9 o'clock in the morning. I'm so sorry for taking a long time. I want to give my highest appreciation to Prof. Tong, all the clinicians, the surgeons here, Prof. Dharmendra, Mr. Azmi Alias, Prof. Azizi Abu Bakar, Puan Shireen, who is a mother in my NTD group. These are all, truly wonderful people who work really hard in keeping the Malaysian population healthy. Thank you, thank you. And any questions, please do ask me. Thanks.
Thank you, Aisha, for the wonderful, wonderful overview. It's fantastic. Always learn something new every time you give a talk. Are there any questions from those who are listening in? Please unmute yourself and you can ask, or you can type in your questions in the chat box or the conversation section. Anybody?
No question, Prof. Tong and Dr. Raisha. It's just Ahmad from HKL here. Just want to congratulate you for a fantastic talk and want to let you know that there's 20 people in this room, all very amazed at the work that you've done and hope that you will continue such great work.
Thank you. Thank you very much for your kind words.
Thanks, Ahmad. Any other questions or comments, or you're too shocked by the information, or?
Yeah, Chung here, pediatric neurology, Again, thank you for the talk. Obviously your passion shone through the talk. It's not 20 participants, but 26 participants heard this talk. So I think you will continue to inspire. Of course, Nature journal is not the own be all and end all. And as you've shown, it's the years of hard work to put in to this. So thank you again for this inspirational talk.
Thank you. I'm open to giving a talk anywhere, really. I just want to see people with spina bifida being looked after in Malaysia, and for us to try to overcome this issue and to protect the womb of the Malaysian woman. That's really my passion. Yeah, that womb needs to be protected. We need to look after our kids.
Okay, any other questions? Let me see, there's a conversation. Okay. All right. Any questions?
Still try and think about this inositol. I mean, is it a completely different pathway to the folic acid? Is there similarities, or is this a completely different
inositol is a simple carbohydrate. It can be found in many naturally occurring foods. It is classically being given to patients with OCD, obsessive compulsive disorder. Nick Green used to joke with me and said, Aisha, if you took inositol, you wouldn't be in such a bad mood every day. So, yeah. But we are seeing many more evidence. You see, with inositol and with folic acid, there's mouse evidence as well as human evidence. All right, it does rescue, and now we see that the inositol pathway is actually married to the formate pathway. So that's another set of mouse that's being looked into. And in fact, vitamin D is also being touted as a requirement in order for the one carbon cycle for the folic acid pathway to work effectively. So, justifiably, I think in order to answer your question, Prof, there has to be like a separate lecture just on the one carbon cycle. Yeah. And actually, to be honest, I am not the best person to talk about the one carbon cycle, but there is a Malaysian, I had a first class biomedical science student from whom I sent. She wanted to do her PhD with me. I said, it's not every day you get a first class. So I wrote to Nick and I said, why don't you take her? And Nick was able to, get the funding, etc. And Yunjin, she published in Nature Communications. For the mouse for the mouse model, the glycine decarboxylase pathway. There's so many great things that's happening within and outside of Malaysia, collaboratively between Malaysians and others abroad. It's just not being highlighted, which is really quite sad, I think. But I think if we were to become a little bit more vocal, I mean, a little bit more brave on social media, we truly need to push the issue of birth defects in our country. I see too many cases, I get too many calls, and one of the things that I'm really fearful of is that there's so many hydrocephalus in Malaysia, irrespective of whether they're spina bifida or not. And this is really worrisome, and I wish there would be more people who would look at studies pertaining to birth defects. But I can tell you honestly, we have applied for 2 long-term research grants. My team is many people. But the grants were written with Prof Tong as the leader of the team, and we failed. We failed to procure 2 consecutive LRGSs, PRGSs, TRGSs. Every year I will try to apply. I mean, people will say, don't complain, Aisha, at least you get FRGS. Yes, I admit I do get FRGS, and I was very lucky to get the support of Emeritus Professor Lam Sai Kit during, especially during my early, those years in when we had a lot of funding money. And that is really, what purchased the equipments in my lab and what keeps us going to this day. But it is an ongoing struggle. I'm not going to joke about it. I worry about my students and whether they're going to end up eating grass. I worry. I worry constantly that they are looking after themselves while doing all this amazing work, for our country.
Okay, well said, Aisha. Well said. Any other comments? Okay, time is catching up. So I think it's already almost 8 minutes past 9. So I think it remains our duty and my great honor to thank Dr. Noraisha for a wonderful talk, and we probably should invite her back again for some other meetings, and she will tell us more about neurotube defects and some of the psychosocial issues, I think, which we didn't manage to cover today. So I think with that, thank you all for joining.